Welcome to NPTEL online certification courses on Pharmacognosy and Metabolic Engineering. This is lecture 58, where under the broad domain of Phenolics, I am going to discuss now the biosynthesis of vanillin in plants. So, the concept to be covered is vanillin and vanillin glucosides, and then vanillin biosynthesis in plants, classical and contemporary views, and then histolocalization of vanillin and vanillin glucoside in the tissue. Okay. So, again vanillin as the structure says so. So, this is 4-hydroxy 3-methoxybenzaldehyde, which is vanillin, and this is originally a C6C1 compound. So, that also produces from C6C3. So, it is all started with this basic phenyl propanoids, and then there will be chain cleavage. So, we are going to see that. So, next let us look into the okay. Now, natural vanillin usually obtained from the vanilla pods. So, vanilla is basically an orchid. So, I will talk about it in the next slide, and from extraction natural vanillin can be obtained, or even from the corn glucose can be utilized through fermentation for the production of vanillin. Other one is that ferulic acid, which is basically abundantly available in the cell wall material and also in bran. So, that can be converted into vanillin because of its structural similarity, as you see here 4-hydroxy 3-methoxy, it is the hydroxycinamic acid, and it is this part is same. So, just this chain cleavage from C3 it becomes C1, and of course, this was acid and this is aldehyde. Okay. Eugenol again was successfully used as a starter substrate for production of vanillin, particularly utilizing different microorganisms, both bacteria and fungi. Okay. So, okay, I will now go to the natural vanillin. So, natural vanillin is basically obtained from the ripened fruit of orchid called Vanilla planifolia. And this fruit is botanically called pod. So, this is what is showing here. It is a green, or rather young fruit. So, this fruit with age converted into a mature fruit, and then eventually it will die. So, the green becomes black in color. So, this is what we call mature fruit, which is now dead. That is why it is black in color. And what happened now, vanillin is there in both the cases. So, now if you take this tissue and analyze, if you make extract and analyze, you will find vanillin not in the free form, but as glucose conjugate. So, this is glucose. Okay. Why it is so? Because it is an aldehyde. Aldehydes are very toxic. So, as it is synthesized in the cytosol, then the plants cannot really retain. So, it immediately try to move it inside the vacuole. So, there will be induction of glucosyl transferase. So, the glucose molecule is added. So, it becomes vanillin glucoside. So, what we write here, this is called vanillin glucoside or glucovanillin. and then when the fruit dies, then what will happen? By the time there will be induction of glucosidase. So, induction of glucosidase, and here we write that induction of glucosyl transferase. And as a result of that, what we see, it produces this glucovanillin, and when glucosidase is induced, then what we will see, here the glucose molecule will be cleaved out. Okay. And leaving free vanillin. Oh, this. I have to draw it once again. So, this is free vanillin. So, the glucose is now out. So, what is available in the market is basically this product available in the market. So, you can buy it in big supermarket, black colored vanilla pod, or it is also called vanilla bean. So, now if you analyze this vanilla bean, you will find vanillin as the major fragrance component. Apart from that, you will find para-hydroxybenzaldehyde, vanillyl alcohol, para-hydroxybenzyl alcohol, and then plenty of other molecules present there. So, that all together constitute the typical fruity vanilla aroma. But again, if you take the smell of this natural vanillin, and if you have the synthetic vanillin available, you will find the difference. Synthetic vanillin is just vanillin, what you get in the ice cream, but the natural vanillin is more of like chocolate fruity flavor, and of course, that is expensive. So, now let us see how vanillin, because of its demand, it is the world's most favorite flavor compound. So, because of its demand and its high cost, because 1 kilo cost more than $4,000 or so, the natural vanillin. So, that is why scientists were interested to understand how vanillin is synthesized in plants, with an aim that they want to do metabolic engineering in the future. So, the vanillin's biosynthesis work started as early as 1970s. By a scientist called M.H. Zenk in Germany. So, Zenk proposed that vanillin can be formed from ferulic acid. So, I have mentioned about ferulic acid in one of the previous slides. So, ferulic acid through a beta oxidation route, it may produce vanillyl CoA, and this subsequently by elimination of hydrogen. So, it produces vanillin, or it can also produce vanillic acid. So, vanillin and vanillic acid structure is pretty same. Okay, I put in this way, CHO OH, OCH3, this is vanillin, and if it is vanillic acid, then this will be replaced by COOH, then it will be vanillic acid, and if it is vanillyl alcohol, then this will be replaced by CH2OH. So, that means all these components are present in the natural vanillin, so which is obtained from the plant sources. Now, after Genk proposed, then there are a lot of work carried out with this. So, like a person from Switzerland whose name is Peter Brodelius. So, Funk and Brodelius so they actually worked during 1990s. So, they have used cell culture of Vanilla planifolia in order to elucidate the biosynthesis of vanillin, and what they found using cell cultures, they are unable to get vanillin. Instead, they found the cell culture successfully produce vanillic acid glucosides. From isoferrulic acid, eventually it produces from isoferrulic acid, and this vanillic acid subsequently form vanillic acid glucoside. So, what happens, this isoferrulic acid is same like ferulic acid. When we draw the ferulic acid structure, so the structure difference is not at C6, but is at C6 position, not at C3. So, in case of ferulic acid, so here is OCH3 OH. In case of isoferrulic acid, the OCH3 will come here and OH will be come here. So, this is isoferrulic acid, remaining part remains same, COOH, COOH. So, this is ferulic acid, this is only the difference. So, Frank and Brodelius could not detect vanillin, and then a group from Japan who is Kanisawa, Kanisawa. So, Kanisawa and his group in 1994, they proposed a different view of vanillin formation utilizing vanilla pod as the source tissue. Okay. So, there but this was published in a conference proceedings, but it never published in any peer reviewed journal. That is why scientists were not very optimistic in accepting their hypothesis. So, now much later a group led by Richard Dixon, so they actually try to elucidate the vanillin biosynthesis in using both cell culture as well as the Vanilla planifolia placental tissue. As a source. So, what they proposed that vanillin is formed from paracoumaric acid. So, all of you know the structure of paracoumaric acid, I am not drawing here. So, paracoumaric acid or 4-hydroxy cinnamic acid. So, paracoumaric acid will make para-hydroxy benzaldehyde and then para-hydroxybenzaldehyde eventually will form 3,4-dihydroxybenzaldehyde, and then so that structure I put it here. So, this is 3,4-dihydroxybenzaldehyde, and it is likely here there is an involvement of cytochrome P450 enzyme. Why? Because para-hydroxybenzaldehyde is only one OH here. So, another OH. So, that is why it is basically a hydroxylase, but this hydroxylase has not been characterized. But interestingly, there is another group from Korea. That group basically isolated 3 OMT from Vanilla planifolia pod, and this 3 OMT converts 3,4-dihydroxybenzaldehyde to vanillin. So, the enzyme is 3-O-methyl transferase. So, that means it requires a SAM. So, here the hydroxyl is replaced with the methoxy, and that is possible. So, this was characterized at the gene level. So, the gene was basically isolated and characterized. Well, now this speaks in favor of para-coumaric acid as the starting material for the biosynthesis of vanillin. Okay. So, this para-coumaric acid will make this para-hydroxybenzaldehyde, and then it should undergo another hydroxylation which makes 3,4-dihydroxybenzaldehyde, and then this will convert into vanillin by a methyl transferase. So, one point here to tell that this step particularly which converts para-coumaric acid to para-hydroxybenzaldehyde, the enzyme was characterized and it was called HBS, hydroxybenzaldehyde para-hydroxybenzaldehyde synthase. So, it was found to be a cysteine protease type enzyme. And it requires DTT for its activation. So, more DTT, more active. But when they have isolated the gene and use the recombinant gene, so it did not really show very much positive result, particularly from the point of this vanillin pathway. So, that is there. So, they basically did not publish this work at the molecular level, rather they kept it at the biochemical level only. They suggested that this is possibly an enzyme, so which is playing important role making para-hydroxybenzaldehyde and which subsequently converted into vanillin. So, this was there. So, the work I mentioned, this was published in Phytochemistry 2002, and this was published in most probably in Plant Cell Report in 2004. So, that was the status. After that, we see a very interesting paper from Denmark. So, where they claimed, so let us go to the next slide. So, this paper appeared in Nature Communications in the year of 2014, and they said vanillin formation from ferulic acid. Vanillin formation from ferulic acid in Vanilla planifolia, and this is catalyzed by catalyzed by a single enzyme, and this enzyme they called vanillin synthase. So, VP van, they have given this vanillin synthase, VP van, and what they proposed that this VP van is basically converting ferulic acid to vanillin, and that let us see how that ferulic acid is formed. So, that ferulic acid is basically forming from this caffeoyl CoA. So, let me, I will not draw the structure because all structures we have seen in the previous classes. So, caffeoyl CoA, from caffeoyl CoA it makes caffeic acid, and from caffeic acid they say it makes ferulic acid, and from ferulic acid vanillin is formed, and this vanillin eventually converted into vanillin glucoside. Now, if you put the arrow, so caffeoyl CoA to caffeic acid basically it's basically the thioesterase. So, thioesterase will release the what it releases the which color I will take, which okay, it releases the CoA thioester out and leaving caffeic acid, and that caffeic acid will be converted to ferulic acid by a COMT, caffeic acid O methyl transferase. So, which basically adds a 3 position a methyl group which makes ferulic acid, and this ferulic acid will converted into vanillin, and this is they have named van, and they also propose that this is likely a hydratase lyase type of enzyme. So, I put here hydratase lyase, and then subsequently this converted into vanillin glucoside. So, if we see the structure of ferulic acid here, okay, let us use another. This will be converted into one intermediate, This is something hydroxyphenyl propionic acid. What we have seen in case of this benzoate pathway, what I have discussed in the previous, in one of the previous classes, and then this will form these are not good at all. So, the enzyme VP means vanilla planifolia, vanillin synthase catalyze both the step. So, the first step is basically a water molecule is added. So, that is why this is a hydration and this is basically cleavage, chain cleavage. So, hydration, what you can also put it here, a molecule of water and here with this acetyl CoA is removed or acetic acid is removed, no CoA is involved. So, acetic acid is removed. So, basically this may be a CoA independent way of vanillin formation, where from ferulic acid it makes an intermediate. So, whenever we put an intermediate, we should put it under third bracket, this means intermediate, and then it immediately converted into vanillin. So, it is basically bi-step enzyme catalyzed by the single enzyme, and subsequently they have characterized this as the molecular level. So, the second one is called retroaldol cleavage and first one is basically the hydration. Now, this paper published in, as I said, it was published in the famous journal Nature Biotechnology, Nature Communications. In the year 2014. Volume 5, article number 4037. This time it is basically totally online journal. So, its article number is there, and this is basically the group head by Birger Lindberg Moller from Denmark. So, University of Copenhagen. So, once this one published, then again the group which led by Richard Dixon from this USA. So, that is Samuel Robert Nobel Foundation. So, and they collaborated with several people in Italy. So, what they did actually, so they again started re-looking the vanillin biosynthetic pathway again, and what we see that they have published a paper in the year 2017, where they say a re-evaluation of the final step of vanillin biosynthesis in vanilla planifolia. And this paper published in again a very good journal which is called Phytochemistry, a traditional journal of its kind, published this sort of work. So, that was published in the year 2017, volume 139, page number 33 to 46. So, what they said basically, they challenge the Nature Communication paper, and they said So, let me read out, you listen. A recent publication describes an enzyme from the vanilla orchid Vanilla planifolia with the ability to convert ferulic acid directly to vanillin. The authors of that paper propose that this represents the final step in vanillin biosynthesis, which is then converted to its storage from glucovanillin by glycosylation. So, the existence of such vanillin synthase, which I said VP van in my previous slide, could enable biotechnological production of vanillin from ferulic acid using a natural vanilla enzyme. But now the point is this, this proposed vanillin synthase exhibited high identity to cysteine protease. So, the cysteine protease I mentioned when Dixon's group in 2002 in the phytochemistry, they said that this is the HBS hydroxybenzaldehyde synthase, which converts coumaric acid to para-hydroxybenzaldehyde, and that enzyme they said cysteine protease. But here also, here also the Nature Communications paper also vanillin synthase what they have characterized, and they said that that is basically showed high identity to cysteine protease. Okay. Now, this current phytochemistry paper, what they said, they reassures that if it is cysteine protease, it really cannot really work with ferulic acid as the substrate. So, it only able to convert paracoumaric acid to para-hydroxybenzaldehyde, not the ferulic acid. So, in other words, if it is a cysteine protease like protein, it cannot convert ferulic acid to vanillin. So, that really is basically they are contradicting or rather challenging the other group, and what they said is this, that the pathway what is operating in Vanilla planifolia. So, it started from the secimate. So, let me briefly put it here. So, okay. So, secimate, which secimate? It should be cafoil secimate. Okay, we start with cafoil secimate, CAFFEOYL. Okay, let me write it in a clear way. Okay, we can start from a little bit of early also, like okay, let us start from this point. Paracoumaric acid or 4-coumaric acid is same, 4-coumaric acid. What Dixon's group proposed, the first author then was Podstolsky. So, this makes 4-hydroxybenzaldehyde, and that subsequently by the action of a cytochrome P450, I will put the enzyme later, which makes 3,4-dihydroxybenzaldehyde, and this one by the action of a O-methyl transferase, it makes vanillin. This I have shown. So, the enzyme what they said that this is a HBS or 4-HBS type of enzyme, and this is a P450, and this is basically a OMT. This has been characterized. Okay. And this is likely to be the route, or the other route what is operating in plants maybe in this direction. 4-coumaric acid will make 4-coumaroyl CoA, and then subsequently it makes coumaroyl secimate, which we have studied, and then it makes cafoil secimate, then it makes caffeic acid. So, if you put the pathway, this makes this, this makes this, this makes this. So, what we know that this is 4Cl, this is HCl, this is C3H, and this is caffeoyl cycimate esterase, which will remove the cycimate, leaving caffeic acid, and this by the action of the enzyme CCOMT, sorry, will make ferulic acid. And this ferulic acid, will be converted into coniferaldehyde, and then by the action, this is by the action of CCR. So, and then this will be converted into aldehyde OMT. So, this will be converted into ferulic acid, and then this ferulic acid to so there is a big question here. So, this one will be ALDDH, aldehyde dehydrogenase. So, the biosynthesis between ferulic acid to fumaric acid is basically through this route, and what they said that ferulic acid may be formed, but this ferulic acid is most unlikely to convert into vanillin. So, what is possible that this CCOMT, this means the caffeoyl cycimate. So, in between it is possible that it may be also that there may be a 4Cl reaction also there. So, in that case from this caffeoyl cycimate from the caffeic acid, caffeoyl CoA may be produced. Caffeoyl CoA can also be produced by the action of a traditional 4Cl, and then this caffeoyl CoA may be converted into ferulic acid by the action of CCOMT. So, in other words, this may not be the case. So, and then it is converted like this. And the caffeoyl-CoA can take part in the formation of vanillin through some undefined routes. Okay. So, which may involve a hydratase, which may involve a dehydrogenase, and which may involve a thiolase, and then finally, there may be a reductase. So, that means what this group proposed, that from ferulic acid to vanillin is not possible, and it should follow a different route what they proposed earlier. So, basically this was a scientific war started between the two group. Both of them have their own views. Now, let us, before I end this class, let me show you the tissue. So, this is again a paper published by this Danish group led by Birger Lindberg Moller. So, this paper published in Plant Cell Physiology, again on this Vanilla planifolia, and this time this was in the 2018 volume 59, page number 304 to 318. So, here basically this is a cross section showing different part of it. So, the mesocarp portion is very thick, and then a endocarp is inside the white portion, and then there are seeds inside, and then placental tissues are also there, and then vascular tissues are also there. So, here what they have done this this desorption, this is a specific mass spectrometric ionization mass spectrometry, and that leads to this picture. So, they have done a longitudinal section, and this longitudinal section they did it by cryosectioning, and then they did directly mass spectrometry of that, and then what you see through this new technique where the red portion is basically the vanillin. So, this is where the vanillin is accumulating, and this green portion is vanillin glucoside, and this blue portion is basically the sucrose. So, they have studied the distribution of this metabolite, that is histolocalization of these metabolites using this high resolution desorption mass spectrometry, and this is really a very nice paper with the modern tools. So, where you could detect the metabolites using mass spectrometry. Similarly, several papers are nowadays appearing in very good journals, where they are localizing the specialized metabolites in the tissue using this technique. Not only that, they have done immunohistochemical localization of VP-VAN. So, that means the vanillaplonia VAN, that is vanillin synthase, exists, and they have they characterize the gene. So, they have the recombinant protein, they raise antibody against that, and then they have used that antibody to localize the protein, VP-VAN protein in the tissue. So, that is what it is. So, what is showing here is again this is a transverse section. So, VP-VAN localization in the mesocarp. So, mesocarp is this the thick portion. So, here is the VP-VAN localizes. and then that is also localized in the plastids, and so that is in brief about the outcome of the latest research, where you can also see the localization of the targeted enzymes. So, this is basically all about the vanillin biosynthesis and the vanillin localization in the Vanilla planifolia. So, the only Vanilla planifolia plant orchid produces vanillin in huge quantity, but vanillin are produced if you isolate the cell wall, you will find and if you do the phenolic analysis, you will find vanillin there, and but that vanillin is basically coming as a result of this cleavage of this coniferyl alcohol or coniferyl aldehyde. So, it is not by the normal route of vanillin synthesis. So, with this I end this class.